Hi there. So in this video, we're going to talk about macrophages. What are macrophages? What do they do? What's their function? So the main function of macrophages that we're going to talk about, starting in this video and in later videos, is their role as phagocytes. They're also going to play a very important role in the immune response by producing cytokines. This video is just going to focus on their role as phagocytes. So, before we go into the functioning of macrophages, let's locate them. Where are they in the body? Well, macrophages are pretty much everywhere um, because their job, right, is to sort of recognize if there's an infection and eat up these pathogens, as well as communicate to other cells that there's an infection. And that's the cytokines part, but we'll get more of that later. So right now I've drawn some structures in the body. I've drawn a blood vessel, I've drawn some tissue, and I've drawn some lymphatic system structures. Go ahead, have lymph vessel with lymph fluid inside, and lymph nodes, and the vessels lead to nodes, which lead to more vessels, which lead to more nodes. So, where are we going to find our macrophages? Well, we find macrophages resident in fairly most all tissues in the body. Macrophages are inner tissues, and they're there to phagocytose, detect, recognize pathogens. They can also clean out any dead and dying cells there as well, but we're focusing on their role in the immune system. So we find macrophages living in our tissues, and we refer to them as resident. They take up a resident there, they stay there, they work there, they live there, they play there. We also find macrophages in our lymph nodes. So you'll notice there in a lymph node, I've drawn a macrophage. So, and this would be any lymphatic tissue like the balt or the malt or the galt, any of those lymphatic tissues that we find, associated with our bronchial or GI tract or mucosal tissues. That lymphoid tissue, it sits right in those tubes. That lymphoid tissue also has macrophages in it. So, how do these macrophages get here? Recall that there are monocytes found in the bloodstream. Never let monkeys eat bananas, monocytes. So, when monocytes travel through the circulatory system, leave the circulatory system, enter the tissues, they differentiate into macrophages. And that's how we get macrophages in our tissues. All right. Oh, what happened here? We have an infection, right? There was some tissue damage. Some pathogen has entered our body through one of our routes on one of our tubes. And now we have pathogens in our tissues. That pathogen might get drained into the lymphatic system, where it goes into our lymph nodes, or this could be pathogens that are being pulled into our lymphoid tissues like the balt, the malt, and the galt. Either way, what's occurring, hopefully, is that macrophages are recognizing the pathogen and responding to the pathogen a number of ways. This video is going to focus on phagocytosis. So, let's visit a little more in depth on how macrophages function, and specifically, we're talking about phagocytosis. So, I'm going to draw a macrophage here, and on its surface, I have drawn a receptor. So, receptors, these are cell surface proteins who function. They're going to bind ligands, and we're going to talk a little bit about what these ligands could be in relation to these receptors that play a role in phagocytosis. So here I have drawn, for an example, the mannose receptor. So macrophages have on their surface a protein called the mannose receptor. What does the mannose receptor do? Well, it's going to bind mannose, found on the surface of many different pathogens. Viruses, bacteria, um, parasites, fungi have the sugar on their surface called mannose. Not all of them, but some of them, many of them. So if mannose is on the surface of a pathogen, and our cells, our macrophages have a mannose receptor, well, mannose will bind the mannose receptor, allowing this pathogen to be taken inside the phagocyte, right? This is a macrophage. And so what's going to occur here is receptor-mediated endocytosis. Recall the terms endocytosis, exocytosis, involving vesicles that are either being taken into a cell or being released from a cell, right? Exocytosis, endocytosis. What's happening here is this pathogens being taken into the cell, into its vesicular system via endocytosis, and it's mediated by the receptor. And so we have a vesicle inside the macrophage, and we can call this vesicle an endosome. Usually vesicles come in bringing cargo, call those endosomes. This is a phagocyte, so we can give this vesicle a special name called the phagosome. And in the macrophage, and we're not going to go into great detail in this video, there are other vesicles called lysosomes. And these lysosomes are enzymes and other reactive species of molecules that are very toxic. And what occurs in phagocytes, such as a macrophage, is the phagosome and the lysosome will fuse together to form the phagolysosome. And those toxins, those reactive molecules, those enzymes will act upon the cargo, in this case, the pathogen, and help destroy it. So that is how phagocytes work to envelop pathogens and destroy them within uh, the phagocyte. So this is all part of the innate immune response. Remember, this is very fixed and very fast acting, right? It doesn't require a whole lot of activation, right? There's the receptor, there's the pathogen binds receptor, boom, in it comes. So can react very quickly um, and very generally, right? Let's talk about another phagocytic receptor, the LPS receptor. LPS, lipopolysaccharide. Um, it is a sugar found on the surface of a number of different bacteria, not all bacteria, but some. So if LPS is on the surface of bacteria, the LPS receptor on the surface of a phagocyte would bind it, bring it into a vesicle, and that's how the macrophage here would help destroy a pathogen that's covered in LPS. Let's get one more phagocytic receptor, and that is the complement receptor. In this instance, we're talking about CR1, which binds to C3b, which is what breakdown complement fragment of the C3 protein, right? C3 breaks down to C3a and C3b. And if that's the case here, CR1 will bind C3b and bring this into the cell. So we know this as an example of opsonization, where the body decorates a pathogen in something like a complement and then makes it more attractive to phagocytes. There are many different phagocytic receptors. These are some examples of them. There are other complement receptors as well, called CR3 and CR4, that play a role in receptor-mediated endocytosis. These receptors bind the IC3b fragment of complement. So C3b can be further broken down into IC3b. We'll actually talk about that in a later video. But those are other receptors that can recognize a pathogen, bring it into the cell. So hopefully you have a good summary of how macrophages, where they live in the body, and how they function to recognize uh, pathogens to take them in to perform phagocytosis.